Welcome everyone to today's installment of the ABRF Virtual Sequencing Seminar Series. We're pleased to have our colleagues with PacBio that'll be presenting today. But before we get started, I just want to share a couple of announcements or reminders for upcoming ABRF activities. First of all, before we get with upcoming activities, please use the chat for your questions. We'll be taking those at the end. So if you'd like to introduce yourself, go ahead and do that. But if you have questions for any of the presenters, please put them in chat and we'll be handling that at the conclusion of the presentation. Also, today's session, as you may have heard, is being recorded. The recording will be sent to everyone tomorrow once it's processed and posted. It'll also be available on the ABRF website. We have the remaining sessions in this series. We're going to take a break next week because we have one of our chapter meetings at the same time. But mark your calendars for the 20th and 27th with two more organizations presenting same time on the same day of the week. And you can see the link to register for those upcoming remaining programs. Many of you know that the ABRF chapters are meeting here in the fall. The NeuralSkid, the Northeast chapter, has their meeting coming up next week, October 12th to 14th in Rochester. There's still opportunities to register, and I believe there are still travel awards available. You can use the QR code here, as well as the link to the travel award opportunity. And then rounding out our chapter calendar for 2022, our Midwest chapter will be meeting the following week, October 17 and 18, in Columbus, Ohio, near the campus of Ohio State University. Once again, you can use this QR code to register. They're finalizing what will be an interesting and engaging program. And then finally, of course, the ABRF 2023 annual meeting. We're terrifically excited to be going to Boston next year in a beautiful time of the year, May 7 to 10. Registration is planned to open by October 31, so it'll be a treat, not a trick. And we have sponsorship opportunities available that you can sign up for already. So with that, I will stop that and turn it over to Sarah, who will can start the presentation and introduce our speakers. So just a moment, please, while I stop the share. All right, Sarah, it's all yours.
Okay, great. Thanks, Ken. Okay, let's get this in presenter mode. Okay, how's that looking? Okay.
Looks great, Sarah.
Great. Okay, great. Okay, so thanks, Ken, and thanks, Anoja, for inviting PacBio to share some updates on our long-read sequencing applications on our sequencer. So we have a pretty packed agenda. I'm joined by 3 other speakers, 2 of my PacBio colleagues, and then Molly Zeller. I'll kick things off with sort of an intro to PacBio, and then continue on to share some updates on targeted long-read sequencing applications. Then I'm going to pass it to Molly Zeller, who joins us from UW-Madison, where she's the lab manager for the DNA sequencing facility in the Biotechnology Center. And Molly's going to continue talking about targeted sequencing, specifically on target capture using PacBio and Twist. Then I'm going to pass it to Liz Sang, who's my colleague at PacBio. She's an associate director in product marketing, and she's going to talk about isoform sequencing and give a little teaser on some upcoming product launch. And then we'll round out the agenda with Mary Jo Galina, associate director of product marketing at PacBio, who is our resident short read expert, and she's going to tell you about our new short read sequencing platform. Okay, so the last 2 years have been really transformative for PacBio and our customers. We've really kind of increased the pace of innovation, and we've released a lot of new products and features that have both simplified and streamlined how you use our technology, but also added a lot of kind of new features and new capability that really expand what you can do with PacBio sequencing. So we've reduced the DNA input requirements and increased the yield on our Sequel IIe platform with new binding kits, new prep kits, and streamlined and simplified protocols. We've really empowered high throughput sequencing with automation protocols, as well as some software updates. Key among those are demultiplexing on the Sequel IIe instrument. And finally, we've released a number of really exciting and innovative new capabilities. In particular, we've enabled 5MC methylation calling on the Sequel2e instrument. We have a new AAV workflow, which enables gene editing research. And lastly, we released the HiFi viral SARS-CoV-2 kit, which enables variant surveillance of the COVID virus. So you may think of PacBio as a long-read sequencing company, but we really want you to think of us as a high accuracy sequencing company. So for the last few years, we've really seen HiFi sequencing transform many areas of research, particularly human genetics, neuroscience, immunology, rare inherited disease research is a major focus for us. We've also had big impacts in plant and animal sciences, infectious disease research, and microbiology. But with the acquisition of Omniome, which is a short read sequencing company, and their highly differentiated and high accuracy sequencing by binding technology, we're now able to push into new markets that PacBio hasn't really played in very much. So cancer screening, cancer monitoring, enabling therapy selection, targeted sequencing of clinical targets and clinical panels, and then also, opportunities for non-invasive prenatal screening. So digging in a little bit on why accuracy matters so much, starting with long reads. So with the length and the high accuracy of HiFi reads, you get the most accurate variant calling and the most complete and high quality de novo genome assemblies. So here I'm showing results from the 2020 FDA Truth Challenge, where we look, the FDA got a bunch of researchers together. They looked at variant call sets, both single nucleotide variants, indel variants, structural variants across the major sequencing technologies, PacBio, Illumina, and Oxford Nanopore, the other long read company. And what you can see here is that when you look at the variant call sets for PacBio HiFi, there's fewer errors with HiFi variant call sets compared to the other competing technologies. In addition, this year, the first complete gapless human genome was published in Science, and HiFi technology was a core sort of required technology for that gapless complete human genome. And also HiFi reads have been selected as the core technology for many large reference genome consortia, like the Human Pangenome Reference, Vertebrate Genome Project, many other large reference genome consortiums. And then just wanted to, dig in a little bit more on the importance of accuracy and the impact that has with this 2021 Nature Biotechnology Technology Review by ABRF, where the authors looked at sequencing systems from a number of different companies, including Illumina, Oxford Nanopore, BGI, MGI, Genapsys, PacBio, Ion Torrent. And what they saw was that HiFi reads provide the lowest error rate of all of the compared technologies in that review. PacBio HiFi had the highest mapping rate and the lowest non-mapping rate. So really giving you the most usable data of all of the technologies. Even in hard to sequence and hard to map regions like homopolymers and STRs, PacBio had the most accurate calls, then the lowest mismatch rate, and then really kind of echoing the result of the FDA Truth Challenge I showed on the last slide. What the authors saw was that PacBio HiFi gave the highest precision in variant calls in clinically relevant targets. So I'll briefly touch on why accuracy matters for short reads. So with the sequencing by binding technology, we're able to give near perfect short reads. So what that means is more than 90% of the bases for our short read platform have quality scores greater than 40. We have a low duplication rate, which means that you throw out less data when you deduplicate your data sets. With the high accuracy reads, we have no index hopping, and you can sequence through difficult and repetitive regions. So really what this means is that there's more useful data with the SBB platform. So we really want to be thinking about the price per answer, not the price per base when we're talking about short read sequencing. So now I'm going to go into the long read sequencing updates. So I'm going to focus in particular on targeted sequencing with a real kind of emphasis on variant detection in humans. So there's a number of reasons to choose hi-fi reads for targeted sequencing. As I've shown you before in the previous slides and others have observed, you get really robust coverage in difficult to sequence and difficult to map regions of the genome. So you can characterize variants and do variant calling over a larger part of the genome. With hi-fi reads, you can get ancestry agnostic discovery of rare or novel variants. You can get high accuracy variant calling. So this would be, not just single nucleotide variants, but structural variants and indels. You can get unambiguous haplotype resolution through direct phasing. And then through our products and in partnership with other companies, we have the capability to do custom panels, which give you really flexible scaling and custom designs. And these are really cost-effective projects. So ultimately, what we want to see is the ability to consolidate legacy molecular workflows like MLPA or qPCR into a single targeted long read sequencing assay. So PacBio has two main offerings for targeted sequencing. It's amplicon sequencing and hi-fi target enrichment. So digging in a little bit on the hi-fi amplicon sequencing, this is a method that you would use. So what you can get with hi-fi amplicon sequencing is full-length sequencing of PCR amplicons. Essentially, if you can amplify it into a single amplicon, we can sequence it full length with one hi-fi read. We recommend PCR amplicon assays for smaller gene panels, 1 to 12 genes. We have supported amplicon protocols using our newest SPK3 prep kit. We have multiple supported barcoding products. We have the barcoded M13 primers. These are pre-mixed primer pairs that in a plate, there's 384 barcode pairs for that solution. And then we also have 96 barcoded SmartBell adapters as well. These amplicon assays tend to be very low cost and very high throughput. There's been a lot of interest, and I'm highlighting some of the recent publications using hi-fi amplicon sequencing. And primarily what we're seeing is a lot of interest in carrier screening, some of the hard to sequence carrier genes like CYP21A2. And then we also see a lot of interest in pharmacogenomics. There's been multiple papers on CYP2D6 and then a recent paper from Stanford on NUT-T15. So shifting to hi-fi target enrichment, this is a new capability that we've built in partnership with Twist Bioscience. So we would recommend this workflow for larger panels, dozens to thousands of genes. This is a real partner play. So there's a optimized Twist long read target enrichment protocol, which is compatible with our SPK3 amplicon protocol to make the SmartBell library. The real advantage of target enrichment workflows is that it's a standard workflow across different panels. So as a core facility, you can get the target enrichment capability up and running in your lab, take customer projects, and they can be looking at different targets and different panel designs, but the workflow is largely the same, it's largely the same independent of the content in that panel. And these are also have a very low cost per sample. And here I'm highlighting the Twist target enrichment protocol, the PacBio SPK3 amplicon protocol that you use to make the SmartBell library. And then we have a best practice guide that walks you through these types of projects. So why did we choose to partner with Twist? Well, there's a lot of kind of technological complementarity between their technology and ours. Twist uses double-stranded DNA probes that have high capture efficiency and exceptional uniformity, which really complements the nice uniform mapping that you get with hi-fi reads. And in partnership with Twist, customers are supported across the entire workflow. So Twist would support customers on the panel design, on the target enrichment workflow and the consumables, and then PacBio supports on making the SmartBell library and the sequencing. So the last thing I want to say is just that we worked with Twist to update their target enrichment protocol. You know that they've been using They've been, they're experts in target enrichment primarily on short reads. So the workflow is largely the same. It's just been updated and optimized for long reads. So the particular design of the panels is a little bit different. We use sparser tiling, so there's fewer oligos to order. You shear the fragments to a larger size, 7 to 10 kb, and we tend to get reads about 5 kb or greater. And we do see better performance in high GC regions, some of these harder to sequence regions with short reads. And so just calling out that we have custom panels available now, so we can connect you with our partners at Twist, and you can get started on designs. As soon as you have a list of genes, you can start working on a design with Twist. So with that, I'm going to pass it over to Molly, who's going to talk about her experience with target capture with PacBio and Twist.
Thank you, Sarah. Yeah, so if you want to go to the next or the title slide, there we go. So my title is Target Capture using PacBio and Twist Biosciences. Just a quick disclaimer, I'm not a bioinformatician, so this is mostly based on how we've actually incorporated this into our lab. And then I do have some QR codes, so if you want to be ready to maybe capture some QR codes for some articles that I'll be sharing, and also a video, just be a heads up to be prepared for that. Next slide. All right, so just a quick little update, or not update, info about who we are. So I work at the University of Wisconsin-Madison Biotechnology Center, and I am the lab manager for the DNA sequencing facility. However, we do have multiple core facilities here, which you also being part of ABRF have probably interacted with some, if not all of us here. And we are in Madison, Wisconsin. And here's just to show you, I do have a nice photo of our team as well as the isthmus here in Madison. Next slide. All right, and then just to showcase what kind of instruments that we do here at we do use here at the at UW, we have instruments from all sorts of platforms. We have two PacBio Sequel 2s. They are named Peanut and Butter, in case you were curious. And then we have a NovaSeq with some iSeqs, and we also do have ONT instruments as well. Our Sanger service has recently just kind of been put to sleep. So we are have actually worked on moving our Sanger service to PacBio. So if you want to learn more, we can chat more about that later. Next slide. So Sarah did actually do a really great job on why we should do target capture on PacBio and what it is about. So I'm probably not going to give you a whole much more information that she didn't already say, because she is the expert and I'm not. But yeah, so we wanted to do this. We have some collaborators here at UW-Madison that work on some really long genes, and using capture on, say, Illumina, it just wasn't quite cutting it. We wanted to check out how we could optimize this on PacBio. And yeah, next slide. And so yeah, so the Twist, we worked together with PacBio and Twist to put together the custom panel of human genes, which I'll get to in just a second here. And then, so Twist designed the panel for us. The total target size was about 1.4 megabases and had just over 1,200 probes. Next slide. So before I get into like our team's actual kind of favorite gene, I just wanted to kind of go through what other genes we did target. So we were mostly interested in NF1, but since we had the option to do a couple of genes all at the same time, we decided to add on a few other lengthy genes. These 4 genes were FLCN, which is folliculin, NF1, OVOL2, and NRG. And in the little table I have up on the top there, that kind of shows the length in base pairs of each gene. And what you'll see is interesting is NRG is a very long gene. And so we wanted to kind of see how much we could get if we targeted it. I mean, what kind of coverage we could get on this gene itself. And then, as Sarah also mentioned, with the T2T project, there's been improvements in the human genome. So I'm hoping that with this improvement, all the progress we've seen, we'll see a lot of improvements in the genome as the time goes on. We were working with human de-identified samples. We extracted high molecular weight DNA. And when I say we, I mean my amazing colleague who's done all of our high molecular weight DNA extraction here at UW. She can make high molecular weight DNA appear out of anything, any organism, I promise. And then we also have sequenced these samples both on PacBio and ONT, and these samples have sequenced fairly well. Next slide. So at UW, we actually work with NF1 a lot. And this is mostly in part because our assistant director of the Biotech Center actually is directly affected because his son has NF1. And so he has teamed up with the UW Center for Biomedical Swine Research and Innovation. And Dr. Deneau is at that center, and they've actually created WMS, which is the Wisconsin Miniature Swine. And it is a pig that is very much similar in size, in body composition to like humans, and also friendlier and easier to work with than conventional breeds. I myself have not had to take a sample from the pigs, but I think it'd be really nice to know that they're much friendlier. And then they're also maintained in a special pathogen-free facility. And I did show an image of the facility also, just to kind of show you the facility, but also just show you that Wisconsin is pretty beautiful. So if you haven't visited, please come visit sometime. Okay, and then so the QR code here is actually just a quick article to kind of showcase the work that we have been doing in the past on this, and also just kind of give you some background on NF1 if you're not familiar with this. So just want to let you know that NF1 is quite common, as it does affect at least 1 in every 2,500 individuals, making it more common than other better known genetic disorders like cystic fibrosis. Next slide. So as Sarah said, the protocol is pretty straightforward. Yep, that's okay. And it's basically, you're just utilizing kind of twist protocol for like their short reads, except it's, like I said, it's optimized to long reads. And so we did shear the genomic DNA down to about 15 kb. And then we did ligate on those adapters, and then ligated PacBio SmartBell adapters on after the target capture hybridization. And I do want to keep in mind that this was our actual first foray at this. We're currently working on more, but did not quite have the data ready in time for this talk, but maybe for ABRF in Boston. Let's go to the next slide. So this is the final library trace. You can see that the average size was approximately 7 kb. And as I mentioned before, we started with DNA that was approximately 15 kb in length. But after prep, our library is much shorter, most likely due to PCR bias or size selection bias. And you can also see that after sequencing, the read lengths are actually closer to 5 kb. And I'll show this again once we get to the next slide, which could just be kind of sort of a sequencing bias. But right now, we kind of just have a lot of theories, and we'd like to replicate this more. And then for those of you that have operated sequels before and you're just kind of curious about loading, I did use binding kit 2.2 primer v4, and the loading ended up to be about 64 picomole. And then you can see here I got a P1 of 86%, which for those of you that know, it's a pretty good P1. Next slide, please. All right, so our results on the femtol pulse, the average size was about 7 kb, which was previously on the previous slide. And then our read lengths did end up being about 5 kb. CCS passing filter was about 45%. And then I do also want to make a note that I did multiplex 8 samples, but we did only get 5 out for the successful demultiplexing. And I think this was due to the adapters that we were using, which is not particular to the optimized workflow as it is now. Let's see. Yeah, so we did also see mapping read percentage about 45% for each sample. And me not being a bioinformatician nor really understanding kind of data analysis, I was like, well, 45%. I was like, isn't that kind of like failing? But after I talked to my boss about it, he was like, no, think about it as like your gene is the football field, and you are capturing something the size of like a yard with 45% of your reads. And I was like, okay, all right, that makes a little bit more sense. So if anybody else needed it be put into different terms, there you go. Let's go to next slide, please. All right, and then, so we did use the CCS reads and no filtering. The default of Q20 was our lower limit. So this is a coverage track of NF1. And I do want to thank Tina at Twist for making these images for me. And then you can also see that there are some valleys that we do have in the in the coverage of this gene, which we're hoping on addressing actually in the current probe set we're using right now, and hopefully will be updated soon. Next slide. And then these are the other 3 genes we worked with. I focused the presentation on NF1 mostly because we have, more of a story with NF1, but we do see a lot, really good coverage on the other genes as well. Still some valleys, but I would say these are really great coverage tracks. Next slide. All right, so after that, after we did the capture, we were looking at kind of like how can we make this better, and how can we help our customers? Being a core, we want to make sure that we get the best results possible. So we're thinking, how do we fix the gaps in coverage? Probably just kind of adjust the probe design, which we can work with Twist at that. They're very helpful in working with you. And then, do we need to decrease off-target effects? Can we get longer read lengths? And then what are the true effects of PCR amplification? These are all just kind of questions that I'm sure we all ask ourselves on a daily basis or whenever you work with the protocol. But just questions we currently have right now. And my last slide. All right, so I did put 2 more, or 1 QR code is actually the same. It's the article from before. If you want to watch a video about NF1 and pigs and the Wisconsin miniature swine, the other QR code is for that video. And then I just want to thank a lot of people, well, the people at Twist, the people at PacBio who have been instrumental and super helpful, and they're my favorite people. Oh, and also the UWBC people who are also very helpful. But my boss, Josh, Emily, who has done all of the extractions and can pull magic out of anything, James, Mark, Chuck, and Danu. So, and then also the Neurofibromatosis Network and all of the NF agencies. With that, I think we are ready to go to Liz, if I'm not mistaken.
Yep.
All right, there you go.
Next slide, please. All right, so I want to give you first a primer for those of you who are not familiar with the PacBio isoseq method, which is using PacBio hi-fi sequencing for RNA sequencing. And let's start with just what it looks like on the bark RNA level. So with short-read sequencing, what you're doing is you generate these beautiful full length cDNAs, and then you fragment them, sequence them with short reads, and try to computationally assemble them. By now, we should all understand that this is highly problematic from a computational. Oh, what's going on?
Sorry, it was advancing on its own. I'll try to catch that.
I think what happens is with the timing, because these are, sorry. It might happen more. So with these short reads, you get only partial views of the isoforms, and then click, please. In contrast, with PacBio hi-fi sequencing, what we're doing is do not fragment the cDNA. Let's keep it full length. And then this way, you can get full length isoform information. No computational assembly is required. Next slide. And what we're offering right now, so you can see there's a little blank. I'm going to talk about what's happening next, but this is what we have today for bulk isoseq sequencing. You could use it for genome annotation of your favorite plant and animal. You can use it for novel isoform discovery in diseases. People have also been using it either standalone or in combination with existing RNA-seq data for differential isoform usage analysis. Next slide. But what we're really interested in going after next is the new frontier in RNA sequencing, which is single-cell transcriptomics. And this is where short reads really fall short, pun intended, because the single cell tag informations are only at either the 3' or the 5' end of the molecules, based on how most single cell platforms work. With short reads, you are fragmenting them and only sequencing the ends, effectively giving you only about 30 to 50 bases of gene level information. This means you're unable to differentiate isoform diversity at the single cell level. In contrast, the same advantage we have with bulk RNA sequencing applies to single cell, where we can sequence the entire molecule from the 5' end to the 3' end, including the poly A tail, and then includes, with high accuracy, the UMI and the cell barcode information. Next. This drives home the point of where we are standing with respect to other sequencing technologies. With short reads, you can only get gene level incomplete information. With other long reads that have lower accuracy, you'll really struggle with identifying accurately the cell barcodes, the UMIs, and also express mutations or variant information that are in the transcript itself. Next slide. So what we are applying now is we are actually productizing. In 1 month, we're releasing this kit called the MOS-seq kit, which is based on the MOS-seq method that's been developed at the Broad Institute. Briefly, MOS-seq is a general amplicon concatenation method. It can be used to concatenate through a programmable series of adapters, creating large inserts. And again, coming back to why can we create larger inserts with PacBio, it's because our HiFi accuracy works very well at 15 to 20 kb libraries. A typical single cell cDNA is only about 1 kb, maybe up to 3 kb sometimes. You can therefore concatenate them into longer fragments and get a throughput increase without increasing the number of smart cells you're sequencing. What the Broad Institute showed in this preprint was that first, in the preprint, they were able to generate 33 million reads on a single smart cell on the Sequel II system. Not only they showed that they can recapitulate the same cell types as the matching short read data on this 6,000 CD8 plus T cell data set, but that also they can see something that you can't see with short reads, which is the lower right-hand corner of showing different isoforms of the CD45 gene. Here, they are coloring the expression of the different isoforms of CD45, such as RB, RAB, and show that they are expressed in different cells. Next slide. So what we are creating and releasing at ASSG in a month is the MOS-seq kit that is the commercialized version of the concatenation method. It is intended to work with the 10x chromium single cell 3' cDNA. It will work for a typical single cell library of 3,000 to 10,000 cells. After the MOS-seq library preparation, it is ready for sequencing. And then we are also releasing a smart link that includes automatic deconcatenation. What the formal word we're using is resegmentation, and also single cell analysis workflow that actually understands isoform classification, not just gene level classification. An expected performance of the MOS-seq kit on the Sequel II system is 40 million full-length reads after read segmentation. You do not need orthogonal sequencing data. No short read is required. And 90% of the reads have accurate barcode recovery. Next slide. Here I'm showing you a teaser, the screenshots from the smart link that we're releasing. So this is a data set that actually we are going to release at ASSG as well. It is on a 4,000 PBMC sample. You can see that there's about 4,000 cells that's estimated using the cell barcode ranking plot. 91% of the reads are in the cells with a mean read per cell of 8,000 and a mean UMI of 4,800. On the right-hand side, we have the transcript summary, where it shows the median genes and transcript per cell, as well as the total number of genes and transcripts identified in the sample. On the lower right-hand corner, for those of you who might be more familiar with long-read transcriptome sequencing, there's a new transcript classification system based on a software called Sconti that describes every isoform against the transcode classification system. Next slide. I think that might be, yeah, that's close to the end of my slides. So just to reiterate, this is the workflow of MOS-seq and also how we're different from other sequencing technologies. It's going to be a single technology run. It will give you isoform information, variant information, and has official software support. Next slide. So you can see that at the end of this year, we will have a new addition to our our RNA portfolio in the form of MOS-seq. Next slide. I think that is the end. Yes, Mary Jo.
Thanks, Liz. I will be talking to you about our short read sequencer. If you haven't heard, last year, PacBio acquired a company called Omniome, and we plan to be the first company to actually offer both long and short reads. And I'm going to tell you a little bit about that new platform, which has not been launched yet. So first slide. So a little bit about the high level overview of the system. It is a mid to high throughput next generation sequencing platform. And really the big thing about this platform is the accuracy. So instead of Q30, which is the norm right now in the field, we can actually achieve a Q40 plus accuracy. And what we intend to do with the system as we launch is to launch other accessories to go along with it, where anyone who uses this sequencer can leverage all of those genomic tools that are available to any short read sequencer that's out there now. So anything that's used like 10x sequencing, the Chromium libraries can be run on this platform as well. So next slide. I'll go into, what does Q40 mean? So I mentioned that the norm right now is Q30. Also, the sequencing by synthesis is the technology that's used for all the sequencers that are out there in the market, not the long read sequencers, but the short read sequencers. So our technology is called sequencing by binding, and it's that chemistry, that upfront chemistry that really confers the accuracy to the system. So here I'm showing Q30s in orange here. And what that really means is that there's a 1 in 1,000 error rate on those sequences. And then in magenta here, the SBB shows Q40s or over, and that what that really refers to is a 1 in 10,000 sequencing error rate. So really we can attain near perfect accuracy, and the error rates they mean a couple things. So they mean not having to sequence at unnecessary depth, and it of course reduces your level of uncertainty because you have lower error rates, and importantly, you're now able to see things, for example, in liquid biopsies, pushing down your levels of detection due to this directly due to the increased accuracy, which is about an order of magnitude better than what's currently out there in the field. So next slide, please. I mentioned that we were launching some accessories products that would go along with the platform to enable some of the third-party tools that are out there for short read platforms. So we'll be in addition to the instruments and the sequencing kits, we'll have library prep kits that use that will create native libraries for a PacBio system. Also other adapters, blockers, and primers, indexing, that's all useful with those library kits. And importantly, we have an adapter kit that a library adapter kit that will convert the P5, P7 libraries into the PacBio adapters, which are AP1. So any library that someone might have in their freezer or refrigerator that has been produced by the vast number of library prep companies, almost all of them have P5, P7 libraries. You'll be able to convert those. It's a fast 5-cycle PCR, and then run that on this platform. It'll be compatible to run. We also have like the blockers and adapters. Those are useful for doing things like ctDNA hybrid capture techniques from Agilent, and so we have all of the accessories that are needed just to plug and play with 10x, with Agilent, and with all of the library prep companies. So next slide, please. Okay, so I'm giving you a quick preview of the system, and then just showing you how we intend to develop it going forward. Our first iteration of the system will be like I said, Q40 quality reads, and we'll be offering a 200 and 300 cycle sequencing kit. So paired end and single end are available to use on this platform. It'll be in the vicinity of 500 million reads. It might be 400 to 500 million reads. We haven't quite locked in on what number exactly it'll be, but right in mid throughput. So you know, about the same number as a NextSeq 550 and a P2 flow cell on the NextSeq, and the workflow is very good. Like I said, that will be the first iteration of the instrument, but going forward, we do see a path toward a higher density flow cell and increased flexibility. So we'll be offering a 100 cycle kit in addition to the other two. We think that we can get to about 2 billion reads, always making that workflow better and easier, more things on board, more flexibility in getting adjusted to a workflow that's already been established. So making lots of changes to make it easier. And then for our next step, we see a path toward 5 to 7 billion reads on it will have to be a V2 sequencer because the optics would need to be higher level to get to see the resolution for that high density of clusters on a flow cell. So just giving you a quick overview of where we're headed. Next slide, please. Right now, we are in beta testing, so we're locking down on a lot of those specs that I already told you about, and having outside labs test them. We have 3 beta testers signed up right now, and we're really taking applications for collaboration. So if you are aware of an application that you think would really benefit from higher accuracy, love to talk to you about it. The ones that we've identified so far are really the liquid biopsy, cell-free DNA or circulating tumor DNA, and maybe like some homopolymers that exist in exome panels. So we have collaboration with Twist, who's validating their exome panel, and looking forward to hearing more about it. And if there is a forum on this this website link here, so if you're interested in talking to me or one of our internal experts about the new platform, please feel free to reach out. We'd love to talk to you. And like I said, we're just commencing beta now, and we are on track for commercial ship in the first half of 2023. Next slide. Yeah, so just a high level product overview. We get 15x higher accuracy. We are experiencing that UMIs are not required. So since the accuracy and quality of the reads is so high, we're seeing that you can get down to very low sensitivities and do not need to use UMIs. And less sequencing. So instead of having, it's relieving you from over sequencing, basically. So some applications that might take you 30x could be as low as 10x coverage to sequence on a high accuracy sequencer. We have seen some excellent homopolymer resolution. So where it's difficult to sequence through these regions with an SBS chemistry, we're finding some great examples where the SVB is able to get right through those homopolymers and have accurate reads through those. And with all of the accessories that we're launching alongside of it, we're really focusing on making this sequencer fit into laboratory workflows. So making a concerted effort to make sure that it's easy to implement in labs that already have some kind of short read workflow, including library prep and data analysis. Next slide. Yep, this I just threw in there kind of at the last minute. So we did some whole genome overview of the actually getting quite crowded, short read sequencing market. So some of the new player, Element and Singular and Ultima are here, as well as NovaSeq, which is the, probably the highest accuracy Illumina sequencer. I haven't evaluated NovaSeq X yet, but I expect it to be right there with NovaSeq. And this is, indels on the y-axis, SNV accuracy on the x-axis, and you really want to be up in that upper right-hand corner. And this is using deep variant as a caller without any training, right? So this is SBS training, actually. So what we're doing right now is we're going to retrain with some of our data that from the SBB platform, and we expect it to be higher and farther over to the right when we do that and recall data. So we're looking like the performance is great, and hopefully showing some really nice data at ASHG after we get some new stuff retrained on that algorithm. So next slide. Yeah, so that's basically it. I'm showing you a picture here. It's a benchtop platform, fits right up on the benchtop. It's about 1.5 feet wide, and pretty straightforward use as a short read sequencer. I think everybody's gonna be able to implement this in labs. Many people are very familiar with the short read processes, and this one's going to be right there. So I think that's all I have. So we'll take questions right now, but if we do not get to your question, please feel free to use the QR code on the left to ask questions and connect with a scientist. And importantly, ASHG is coming up on the 25th, I think it's 25th through the 28th of this month. And I would love to meet with you if you have any questions about short reads. And of course, all the other speakers here are happy to find you there. So scan the QR code on the right, and you'll see our program and sign up for content. You can actually, if you don't plan on going to ASHG, you can get a recording of our workshop, which should be very exciting. We're making some really interesting announcements there. So I suggest you don't miss it. But we can take questions that are in the chat now.
Liz, do you want to come on and maybe read some of the questions out?
Yeah, so I can answer the first question. For when doing MOS-seq, do you have to run short read RNA library? So do you have to run the single cell library on short read sequencer as well? The answer is no, no, no. Hopefully that's clear. And the question from Molly, can you say something about the transition from Sanger to PacBio?
Yeah, I can happily, I can say that we're having a lot of success. We're still kind of like early stages, but basically what we did is we just transitioned everything that we put on Sanger, and we're kind of just pooling it all, putting it on a smart cell. I'm really hoping to have more information by ABRF 2023 to present, and I don't know if I can say a whole lot more at this moment in time, but I would love to.
Molly, can you comment on how you're doing barcoding and pooling? Like, what are you using to barcode the samples?
Yeah, right now we're using just the 96 PacBio adapters, not the ones that you guys made, but the ones like you had a list of beforehand.
Okay.
That we ordered from, I think, IDT. But yeah, so we're actually going to be creating some more of those, and then kind of also having like a super multiplexing kind of scheme. So
how high do you think you need a multiplex to make it kind of cost effective, or just production scale?
Even 96 isn't too bad. It's definitely not going to be like Sanger prices, because that's, I don't know, it's just, but also it's going to be more accurate, it's going to be better data. But yeah, I think we're going to try to aim for like 384 at least, but 96 is still at least pretty cost effective at this point in time. So
cool, thanks. Any other questions from the field? You guys can put them right in the chat. Okay,
Well,
maybe
Sarah,
we
perhaps
should.
we covered everything.
Okay, great.
So thank you all. Thank you very much to our friends at PacBio. Thank you to Anoja Herrera at Stowers for organizing this terrific series. As I mentioned, next week is sort of an off week. We won't be having a seminar at this time. If those are available, please join us at the NeuralSkid meeting and as well as the upcoming Midwest meeting. But the seminar series continues in 2 weeks, October 20, also at 2 p.m. Eastern, for another presentation. So once again, thanks everyone. We really appreciate it. And as I said, look for the link to this presentation, the recording of the presentation you'll receive tomorrow in your email. Thanks again, everyone. Appreciate it. Have a great day.
All right,
Thanks,
thank
everybody.
you.
Have a good one.
Thank you.